Hello everyone, my name is Emma Thrift and I'm a second year PhD student in Dr. Jennifer Tank's laboratory at the University of Notre Dame. Today we'll be talking about antibiotic resistance and what this has to do with streams. Bacteria are present everywhere, in our air, soil, our water. When our livestock get sick from this bacteria, many farmers will treat the animals with antibiotics, the same types that you or I will use when we get a bacterial infection. But here's where it gets a little scary. When antibiotics are added to livestock food, the animals, say a cow, will excrete it in the form of feces. But these antibiotics are pretty much in the same form as when they consumed them. So this means they're still working and they have a chance to interact with all of that bacteria in the soil, air, and water. The antibiotics kill the weak bacteria, but leave the strong and adapted ones behind. This leaves an army of resistant bacteria that have the potential to infect you and I. So what does this all have to do with streams? Well, when it rains, the water washes the manure off of the farm fields and the water ends up in our streams, along with the antibiotic-resistant bacteria and the genes that are inside of them that end up encoding for resistance. So here's where my research comes in. We don't exactly know how the nutrient-rich agricultural waters travel through these streams. There has been evidence that biofilms, or the algae, bacteria, and fungi that grow on the stream rocks and sediment and make the rocks really slippery, can use this genetic material in the water as a nutrient source. Yum. So what happens when this biofilm is already bathing in nutrient-rich agricultural streams? Does it still absorb resistance genes easily at the same rate? We don't know. To find out, we are using 24 mini recirculating streams at the experimental mesocosm facility at the University of Notre Dame. We combined various levels of nutrients and shading treatments to these streams and added antibiotic resistance gene-rich cow poop to each of them. Then we take water and biofilm samples over time to see if the biofilm removes these genes at a faster or slower rate depending on the treatment. Cool, huh? I'm hoping that this research will give us insight into how these genes travel through our waterways. This will be key to understanding how we can manage our lands to reduce the risk of antibiotic resistance. Thanks for listening, and I hope you find this research as cool as I do.